Welcome back to our video on population genetics. This is part 2. When we left our story, we were about to tackle each of the 5 Hardy-Weinberg conditions, seeing what happens when we violate these conditions, how it leads to changes in the gene pool. Let's start with the first condition. No mutations can occur. Well, why not? How does that change the gene pool? Well, this is probably the easiest of the 5 conditions. What if mutations do happen? For example, what if these two white alleles mutate into brown alleles? Or what if these brown alleles, what if there's a new mutation in the population and we end up with some black rabbits? That's going to directly adjust these allele frequencies and therefore the genotype frequencies. So that's a pretty easy one. Let's move on. Now, the population being large, I'm actually going to do that one last because it's the one that's going to take the most work. So let's skip to mating is random. For the Hardy-Weinberg condition to be true, we have to have no sexual selection. Mating has to be random. And there are populations where mating is random. Think about flowers releasing pollen into the air. That's pretty random mating. But there's also animals that engage in random mating. But let's look what happens if we have what we call sexual selection. So here's our female rabbit, and here's 2 male rabbits that she can pick from. And if she makes a conscious choice, maybe she believes that the brown rabbit looks healthier, that the brown coat color represents a better nutrition or better genetics, then she might choose to mate with brown rabbits more frequently than with white rabbits. And that's going to adjust the allele frequencies in the gene pool as we get more brown alleles passed on rather than white alleles. So that one's also pretty easy. How about number 4? No individuals may enter or leave the population. No immigration out, no immigration in. Well, again, not a hard thing to understand. If I have a rabbit, let's say this rabbit, in some populations where as the males, when they get older, they leave the group and go out to find their own group. Maybe this rabbit's just fed up with it and is ready to leave. Well, when he leaves, he takes his alleles with him. So they're no longer in the gene pool. And that's going to pretty directly change these frequencies. And conversely, if someone enters into the gene pool, they bring their alleles with them and add them to the genetic content of the population. So that will obviously change the content of the gene pool. How about number 5? This is an interesting one. We've looked at natural selection before, but this is looked at in a slightly different way, looking at how it impacts the alleles in the population, the gene pool. So let's say this is our environment, and we'll populate our environment with brown and white rabbits. Well, sometimes some genotypes are better suited for survival. In fact, many times they are, especially in this case when we'll bring in a predator here, a hawk. And in this case, we might see that over time, more of the white rabbits are predated on, and therefore the population shifts and more brown rabbits make up the population than white rabbits. But if the environment were to be changed a little bit, we could see where this would drastically change the outcome, so that over time the number of brown alleles would drop in the population and the number of white alleles in the gene pool would go up. So natural selection affects gene pools. Well, now we're ready for the last one, the one that's going to take us a little more work, and we're going to let it be an example of how we do what we call Hardy-Weinberg problems. So Hardy-Weinberg conditions say that for populations to maintain their genetic equilibrium, for things to not change, the population has to be large. What happens if it's not large? What happens if it's small? Well, let's do a comparison. Say we have population A with 1,000,000 rabbits and population B with only 1,000. But we do some sampling, we find out that both these populations are the same, meaning they have the same basic makeup, and 91% of the rabbits in this population are brown rabbits. Can we, for this population, determine the allele frequencies for both these populations and the genotypic frequencies? Well, what do we know? We know that 91% of the rabbits are brown. Does that mean that 91% of the alleles in the population are brown? Well, no, because not all brown rabbits carry just brown alleles. Some of them are heterozygous and carry a white allele because the brown allele is dominant. So I can't look at one of these rabbits and see these white and brown alleles that they carry, so I'm kind of stuck. But what do I know? I want you to pause the video now and try to think about what you do know that might help you get started in this problem. Really, pause the video. Some of you have a lot of experience doing these problems, and I want to see if you can do it before I tell you the answer. Well, if 91% of the rabbits are brown, that means 9% of the rabbits are white. And the only genotype that can show white rabbits is homozygous recessive. So if 9% of the rabbits are white, then 9% of the genotypes are b homozygous recessive, which we said we can represent by q squared. So we know q squared is 0.09 or 9%. Well, what's the next logical step to fill out all these factors? Pause the video again and see how far you can get. Well, hopefully you realize that you could take the square root of q squared and find q, and so that q would equal 0.3. Be careful, it's not 0.03, it's 0.3. And so the frequency of the recessive allele, B, is 30% or 0.3. Well, we also know that P plus Q has to equal 1, and we know that Q is 0.3, therefore P has to be, that's right, 0.7. So we can put 0.7 in here as P, the frequency of the dominant allele. Well, what's the next step? We can square P and find P squared, and P squared is 0.7 squared, which is 0.49. So for our homozygous dominant genotype, P squared, the frequency of P squared is 0.49 or 49%. And all we have left to do now is find 2PQ. 2 times P is 0.7 times Q, 0.3, 3 times 7, 0.21 times 2, 0.42, or 42% of our rabbits are heterozygous. Now, using the Hardy-Weinberg equations, we have given ourselves a mathematical image or a mathematical model of what this population looks like. We know the makeup of the gene pool, and we know the makeup of the population. So here's the question, or let me give you a question of practice before class. Using this population over here, if Hardy-Weinberg conditions are held true and you revisit this population several years later, when the population has grown to 1800, how many brown rabbits would you expect to see? Now, I'm not going to work that problem, but I want you to work it and bring it into class. Show it to me for maybe a little extra credit or something. Maybe. We'll see. But just challenge yourselves. I'm going to go back to our question. What's the difference between what happens in this large population versus what happens in the small population? Well, we need a random event. How about a landslide? Let's say that one day your rabbits are out getting rabbit food and doing rabbit things, and there's a landslide that happens by chance and dumb luck to kill 100 rabbits, and 50 of those rabbits that were killed happen to be white. Recalculate the allele and genotypic frequencies for each of these populations. Pause the video and see if you can't work it out. I'll start with the smaller of the two populations. Originally, 9% of the population were white. So 9% of 1,000 is 90. And we lost 50 of those white rabbits to this horrible landslide. So we're down to 40. But we lost 100 total, so we divide 40 by our new population size, 900, and we get 0.044. And that's our Q squared. That's our frequency of white rabbits, our frequency of homozygous recessive. And notice we went from 0.09 to 0.044, which is about, we've cut it in half. And then we take the square root of Q squared to find Q as 0.21, and we subtract from 1 to find P, 0.79. And we can see that we've drastically changed our allele frequencies almost by 9%. Now, let's see what happens when we do the same calculations with our large population. Again, originally 9% were white, and 9% of 1,000,000 is 90,000. Well, 50 of those poor buggers got killed in the landslide, so now we're down to 89,950, which we divide by the new population, and we see that our Q squared is 0.808995, which is essentially the same as 0.09. And if we take the square root of that, we find that our Q is 0.2999, which is still basically 0.3, which makes our P 0.7001, which is basically still 0.7. So the point is, it didn't change. The large population was not affected by the random event, whereas the small one was. It's the exact same event. The exact same event had a different impact from one population to the next, and we call that genetic drift. The change in the allele frequency in small populations due to random chance events. That exact same random event has basically no impact on the larger population. Now, genetic drift is a concept we'll probably talk a little bit more about in class because sometimes students have a hard time with it, but two examples are bottlenecking, which is what we just described. Basically, our population was culled, was brought down by a random event, and the gene pool of the survivors is different than the original population. We also have one called the founder's effect, when a new subpopulation is established and the founding population, by chance, has a different gene pool composition from the parent population. So there we go. We have our Hardy-Weinberg principle, our 5 Hardy-Weinberg conditions, and our Hardy-Weinberg equation. We're going to be using this a lot in class to solve problems, so be ready to do some problems when we get into class. And that brings us to the end of part 4 of evolution, population genetics. Leave me any questions you have in the comments below, and I hope you learned something.